Welcome, everybody. The Office of Programs to Enhance the Neuroscience Workforce within the National Institute of Neurological Disorders and Stroke is very pleased to welcome you to our December Open Stage webinar. This is a part of a webinar series that we do at the targeted outreach effort for our diverse neuroscience community to learn about NINDS programs and kind of meet the people behind NINDS. This month, we are really excited to introduce kind of behind the scenes of NINDS Intramural Research Program. Before we get started, I just have a few housekeeping items. So please note that all attendees are muted, so this is a webinar. You are very welcome to use the chat, but if you have a question, please do submit it using the Q&A box. You can ask your question with your name, or you can ask anonymously. This presentation is going to be recorded. It's going to be archived on our website. After the webinar, I will send an email to all registrants with the contact for the panelists, with the slides, and with a whole bunch of resources. So don't worry if you miss something. I'll share the slides, I'll share the recording, I'll share all the contacts. So you can have all that information in your inbox. And then also if you need a transcript, that is available at the bottom of your screen. So again, please do stay connected with us. We do have these webinars about monthly, so we hope you join us for the next one. And with that, I'm very excited to pass it off to our Scientific Director of the NINDS Intramural Research Program, Dr. Jeff Diamond. And actually, before I do that, I'll just mention the agenda for today. Dr. Diamond and Dr. Plant will give a brief introduction to the Intramural Research Program. Then Dr. Plant will introduce our intramural investigators, and then we'll do kind of a fireside chat and Q&A. So sorry about that. With that, I will now pass it off to Dr. Diamond to get us started. Thank you.
Thank you, Anna. Thank you, everybody, for coming today. I should first thank Anna and the Office of Programs to Enhance the Neuroscience Workforce. They do a lot of really good work on behalf of our institute and the larger neuroscience community, and I really appreciate all the hard work that she's put into making this happen. We know from the registration list that there's a lot of people tuning in from a lot of different areas from within NIH, but mostly from places outside of NIH at any of a range of steps in their professional careers. And so I'm going to start out by assuming that you all don't know much at all about NIH or the intramural program. And so for those of you who know a lot, I apologize, but listen along and maybe you'll pick up something that you hadn't known before. What I want to start off with is something very basic, which is the mission statement of the Institute. And it's a pretty, on the face of it, a pretty simple statement. And that is the mission of NINDS is to seek fundamental knowledge about the brain and nervous system and use that knowledge to reduce the burden of neurological disease for all people. It's a pretty straightforward sentence. I put it up here because it has some very important parts. It's very intentionally written, and it includes some things that show important aspects of the intramural program. First of all, this first part about fundamental knowledge and what we call fundamental or basic research is a really important part of the NINDS portfolio overall, and certainly the intramural research program. And that is understanding that the building blocks of the brain, the molecular, the genetic, the cellular processes that underlie the function of our nervous system. This is a key part, and it plays into the next part, which is to use that knowledge to reduce the burden of neurological disease. Now, the brain is fascinating in its own right, and studying it just for that purpose is a noble thing to do for your career. But at NINDS, we don't stop there. We need to translate that fundamental knowledge from the bench to the bedside, to turn that knowledge into therapies and treatments that can benefit people who are suffering from neurological disease. And finally, and this is as important, this is for all people. We recognize the diversity, the genetic diversity and of the people who are suffering from disease, and we need to take that into account when we develop treatments. So this is, a really important thing, this mission statement, and it includes a lot of, really important aspects of what we aim to do. So this is an aerial shot of the campus, the campus of the NINDS Intramural program in Bethesda. It's a big place. There are more than 50 research buildings across this campus, over 1 million square meters of research and clinical facilities. That's over 10 million square feet. And there are over 1,200 laboratories in a range of different institutes that make up the National Institutes of Health. And a large part of that community is devoted to the study of neuroscience. And not all of those, that neuroscience is happening in NINDS. There are about 180 labs on the intramural campus that are studying neuroscience. And they represent 14 different institutes. And so you can see a range of the institutes that study neuroscience, NINDS up here at the top being just one of them. NINDS has, within its intramural program, has about 45 labs. It varies a little bit from time to time, but performing basic or fundamental research, translational and clinical research. And these are the three of those many buildings. These are the three buildings on campus that house the NINDS faculty. So the research within NINDS and across the neuroscience community, across the whole campus. And I want to stress that because if you're thinking about coming and studying at NIH and you want to do neuroscience, we like to think that NINDS is the best neuroscience-oriented institute, but it's certainly not the only one. There's a huge range of work being done, and I've just put some images up here to show the range of things that are studied. There's a lot of study on the genetics of basic cellular functions and how those genetics relate to disease. Mapping neural pathways, understanding the very basic structure function relationships in individual proteins and also in brain circuits, understanding the basic features of synaptic transmission, the way that neurons communicate with each other, how the brain develops, and a range of technologies that allow us to perform gene therapy or to use advanced imaging techniques to diagnose and treat disease. One of the really great things about NINDS is that we have a terrific training office and a vibrant training program that is directed by Dr. Faith Harrow Plant, who is our Director of Training and Education. And I'm very pleased that Faith is here, and I want to hand it off to her so that she can talk more about the training opportunities that we offer. Faith?
Thank you, Dr. Diamond, and hello, everyone. First of all, I too would like to thank the Open Office for inviting me to be able to tell you a little bit about the intramural training program here at NINDS. So I am the director of the Research Training and Career Development Office, and my team is responsible for helping our early career scientists or trainees, as we call them, navigate and make the most of their training experience while they're here at NINDS, and also helping them to transition to the next stage of their career or education. So this slide here illustrates the many research and clinical training opportunities available at various stages of education at NINDS. And that starts from high school seniors who are eligible to participate in our summer internship program, to college students, to recent college graduates, PhD students, medical students, and we do have postdoctoral fellowships and a number of clinical fellowships and residency programs. If you have questions at any time about any of the training programs at NINDS, feel free to email us at dirtraining@ninds.nih.gov, and I have it in the chat so you can take that down. And again, if you have any questions at any time, feel free to reach out to me. Okay. All right, so who are our trainees at NINDS? At any given time, this is what our trainee population looks like. In the summer, our population grows. As you can see, we have about 70 more trainees with the arrival of the summer students in the summer. And what I hope you can appreciate from this slide is that by coming to NINDS, even just as a summer student, you get a chance to interact with and learn and engage with all the other types of trainees at the NIH and decide, if this is something that you would like to pursue further, if you'd like to return in one of these positions. And you also get to interact with our wonderful investigators and scientists and start thinking about your career beyond training down the line, if this is something that you would like to do in the future. Okay, so let's start out with the summer internship program. I'll just tell you about a few of these programs, but the summer internship program is a full-time 8-week minimum paid internship conducting research in basic or clinical or translational neuroscience in our labs in Bethesda, Maryland, under the mentorship of leading neuroscientists. As a summer student and as any trainee, you have access to comprehensive career development resources. And if you're interested in applying to our program, the application period opened just before Thanksgiving. You have until mid-February to submit your application. However, I will give you an insider tip. You do not want to wait till February. It's on, we accept students on a rolling basis. And the earlier you get your application in, the better, and that increases your chances of getting admitted to the program. If you are interested in getting more information, again, email us at that email, dirtraining@ninds.nih.gov, and we will be happy to guide you through the application process and give you more information about NINDS investigators, NINDS labs, and just some tips on applying and submitting a successful application. Okay, so like many institutes of the other institutes at the NIH, NINDS has a cohort program within the summer internship program, and this is called the Health Disparities in Tribal Communities Summer Internship Program. The program was developed out of the recognition for the need of for diverse perspectives in neuroscience research, and we aim to recruit a diverse cohort of students each year. In the HDTC program, our students engage in weekly activities that focus on health disparities research and also highlights the importance of scientists from all backgrounds participating in research. And again, if you're interested, you could reach out and get some more information. The other program I would like to highlight for our college students, our soon-to-be graduates, is the postdoc program. So the postdoc program is a program for recent college graduates where they spend either 1 or 2 years at NINDS in preparation for transitioning to the next stage, which is often grad school, med school, MD, PhD programs, or other types of professional schools. So if you're interested in that, reach out to me and I can give you more information. And the last program I want to highlight today is the Graduate Partnerships Program. The NIH is not a university, but we partner with universities across the country and actually internationally, where our graduate students fulfill all the requirements for their grad program at their home institution, but they conduct all of their research at an NINDS lab. And as I mentioned earlier, we have a number of clinical fellowship and residency programs. We have postdoctoral fellows from within the US and across the globe. And in the interest of time, I won't go over these, but if you would like more information about these or any of the programs, again, send us an email and we will be happy to answer any questions you have. So with that, I will turn it over to our first panelist. We're excited to introduce our panel of intramural scientists. You will see that our panelists represent the full spectrum of careers at NINDS, from a trainee all the way through a senior investigator. And you'll also see that our all the types of research are represented. We have basic and we have clinical research. And with that, I will turn it over to our first speaker, Hector Cancel Asensio, who is a clinical research postbac, IRTA, that stands for Intramural Research Training Award Fellow, and he is in the translational neuroradiology section. So Hector, I'll turn it over to you now.
All right. Hello, everyone. My name is Hector Cancel Asensio, as Faith said, and thank you so much, Faith, for that introduction about the postbac program. I'm going to be talking a little bit about my journey and also my postbac experience and journey as well here at the NIH, at the NINDS as well. Well, I try to not overshare a little bit, and I'll keep it simple. I was born and raised in Puerto Rico. I come from a huge and humble family. Back then, my grandfather, I guess they needed manpower in the farm, and they had 10 children, including my father. And I have, on my mother's side, I have two uncles, but my uncles have over 12 to 15 cousins. And as you can guess, up to this day, I still meeting new cousins, new family members at the gatherings. At the left side is my father's family side, only the uncles, and on the right side, my mother's family side. As I gain consciousness, I always been athletic and playing sports, and I got the privilege to play for the baseball team in the University of Puerto Rico, Bayamon. That's where I did my undergrad in human biology, and also I did the track and field team. And it was amazing be able to be part of a team player, learning how to build and have different type of backgrounds and ethnicities here on those teams. But sadly, 2 months after starting my first year of college in 2017, as you might be familiar, Hurricane Maria hit Puerto Rico, a deadly Category 5 hurricane, but it bring out the community of Puerto Rico all together. We were able to help each other. We spent over 3 months without electricity and portable water. It was a really rough journey, but this adverse event helped us a lot to be resilient and move on. This also worsened Puerto Rico's economy, and then scholarships were shut off, sadly. So baseball and track and field was something I was not able to continue, even though I wanted, but I had to seek for jobs. And that's when I moved into doing more research. Into those years, I did a little bit more of animal research in the beginning of my journey, but then I got opportunities to do also some shadowing, some clinical research, and some benchwork research. That's when I started to start feeling I was a little bit more into clinical research and get also a more taste of what is the science work that we can do in science and research. And everything seemed to be coming back to normal. But then, as we all know, what happened, the COVID-19 hit all the world, and everyone was, we were all lost. We didn't know what was the future going to be headed. And I got lucky enough that there was an opening in the Puerto Rico Comprehensive Cancer Center. And I started doing also more clinical research there. And I was able to be impactful in many, many ways, not only in the clinical way, but also in our population in Puerto Rico. And yeah, so this was an amazing experience. This is when I started to understand more about the opportunities that not only the science can bring us, but being educated and following up into programs can help you. I did research here and I enjoyed it. And with the hopes of strengthening my application, I then graduated. As you can see here, that's my lovely family back in my graduation into 2022. And that was June 2022. On September 22, I joined the NIH as a postbac in the postbac program. And it was very amazing experience. Sadly, not even a month within my acceptance into this program, I received the notification that my father got diagnosed with stage 4 liver cancer. You can see my father is on the left side on the picture. And that first year, I was here, but not here. As you might experience is and we as scientists, we can understand that the field and a diagnosis like that is going to be a very hard one. So I could see maybe that I had to give more to my family at that moment. I talked with my PIs, my coworkers. They were very supportive. After the days, they have been very supportive with all the process. And I almost travel every other week to Puerto Rico, and I kept balancing the research as better as I could with the resources as I could. And the NIH also has a lot of resources. I also seek a wellness advisor. I use all the tools I can to become a better person in the future. And sadly, less than a year ago, he passed away after he diagnosed. But thanks for the help of not only the NIH, but my coworkers in the lab. I kept my journey. I seek opportunities, participating in conferences, demonstrating my research, being involved in a future generation at the NIH. That's another thing. So planting seed into our future generation here at the NIH. I try to be as active as I can. I also participate in the SACNAS as a board member, and I also volunteer for the NIH Catalyst as a writer. So the NIH has very, very good opportunities, brings a very broad perspective as well. I've been able to work with multiple different researchers, clinicians, fellows, and I do research in the neuroimmunology clinic lab with more focus into the multiple sclerosis condition. I do patient evaluation measures. And right now my project consists of measuring the thickness of the retinal layers thickness with optical coherence tomography or OCT scans, and correlating it with the relationship with the cortical lesions found in the brains with the MRIs. Now, in the waiting game, that's a conference right there. And now I'm in the waiting game for the medical application. I cannot be more grateful, not only for the resources, but for the support I have received from the NIH about the experiences and opportunities that, Puerto Rico is a small island, and I would never thought I was going to be even talking to all of you. There's a lot of things I will be able to cover, and feel free to reach out if I could be a help and a tool for guidance onto the postbac assistance. And I think that's going to be a little bit of my journey. I hope you enjoyed, and thank you.
Thank you, Hector. Thank you so much for sharing your journey with us. We really enjoyed that. And if anybody has any questions, remember you could put them in the Q&A and we'll get to them at the end. Okay, our next speaker is Dr. Jean Biou. And Dr. Jean Biou is the head of the Global Neurology Program at NINDS. So I'll turn it over, Dr. Biou.
Thank you so much, Dr. Plant. As Faith so graciously introduced me, I am Jean Biou. I am a neurologist with specialty in neuroimmunology and neuroinfectious disease training. I'm currently what we call a staff clinician here at the NIH, and I'm head of the Global Neurology Program. And I'll briefly just kind of detail how I got from here to there. So I grew up in Louisiana. Oh, my little slides are slow to come. Here we go. There we go. Sorry. In an area of basically the swamp, in an area called the Atchafalaya River Basin. I stayed in state for college for my undergraduate degree, and of note, I got a liberal arts degree before going to med school. And I went to medical school at the Chicago Medical School at Rosalind Franklin University. After freezing for 4 years, I decided I maybe didn't want to stay there, but in med school, I became greatly interested in neurology. And after doing an intern year at the University of Chicago, I made my way to Baltimore, a little less cold, where I did my neurology residency at Johns Hopkins. So I had a wonderful time at Johns Hopkins, but while there, I really found that I was very interested in kind of the weirder cases of, weirder patients, patients who may not have had an obvious diagnosis. And these ended up being patients who had either infectious diseases or inflammatory diseases or what we call neuroimmunological diseases. And so I started looking around for further training because I just wanted to specialize in some of this fascinating field. And when I was looking for fellowship opportunities at that time, especially, I really found that if you wanted to study either neuroimmunology or neuroinfectious diseases, you were kind of shoeboxed into studying either MS, doing an MS fellowship, or a neuro-HIV fellowship. That's changed to some extent, but there was one place that was very different, and that was the NIH. So, the NIH had this really interesting clinical neurology fellowship that was in neuroimmunology and neurovirology. And so I became interested in that and did a little research project during my residency, just to see if I thought, if I liked the area, it's not too far away from Hopkins and that sort of thing. And I found that I really loved the environment. I thought it was, going to be a great place to do further training. So I did end up signing up for a neuroimmunology and neurovirology fellowship, partly because, there were lots of different reasons. But part of this was because I really wanted to explore the opportunities that the NIH had regarding research. I had done some research up to that point, but didn't really know if research was the right fit for me. So I ended up doing some research on a number of different viruses, including human herpes viruses such as HHV6 and EBV, and how they play a role in different neurologic diseases such as multiple sclerosis and epilepsy, as well as HTLV1, a type of retrovirus that causes a disease affecting the spinal cord called HAM-TSP, or HTLV1 associated myelopathy and tropical spastic paraparesis, which is a quite rare disease. And through this fellowship, I was able to do all sorts of different kinds of research. I was able to do research at the bench, developing some PCR techniques, as well as clinical research, seeing some of these, HAM-TSP patients, doing small clinical trials, seeing how medications might affect their disease progress, and kind of that in-between type of research, which we call translational research, where we, taking, the specimens from these patients, bringing them back to the lab and doing research on them and things like that. Stuff that really, it's hard to find this kind of an environment where that type of research is really possible. After a couple of years of my fellowship, I started looking for a new started thinking about what my, job opportunities would be like outside of the NIH. But that's about the same time that the West African Ebola epidemic happened, or outbreak happened in about 2014, 2015. And I think I was just at the right place at the right time, where, our Infectious Diseases Institute, NIAID, was doing a research project on Ebola survivors and looking at kind of the natural history of how these survivors did. How do they do long term? What kind of issues do they have? And they realized that there were some neurologic issues involved. And I got kind of roped into this project, thankfully. And it really became a big project for me, where I started looking at, how these survivors do long term, doing careful neurologic evaluations over time. And that I also became involved, and this was mostly in Monrovia and Liberia and West Africa. But I also became involved in a number of other projects related to NIAID's efforts across kind of different areas of the globe. And through all of this, through all of these projects, this kind of ended up leading to the development of this new program called the NINDS Global Neurology Program. We officially started this in 2022, and our mission is to conduct international clinical research of neurologic disorders with the focus in neuroinfectious and neuroinflammatory diseases in lower middle income countries, and to provide training in international clinical research as well. So my main project that I have done, over the past 10 years or so has really related to the long-term neurologic sequelae in Ebola virus disease. Since it's such a rarely studied disease, it's kind of just a unique opportunity that not many neurologists especially have been able to do. And so here are a few pictures related to that project. I have all the way on the left, I'm with some of my Liberian colleagues. Right next to that, we have a picture of the CT scanner that we were able to install in the capital city of Liberia in Monrovia at the main hospital, JFK Medical Center. And it was the only CT scanner in that city at the time. And it has really drastically changed how their clinical care has been, even though this CT scanner was initially put in for research. Next to that, I also have a picture of me all dressed up in my PPE, or my personal protective equipment, when I was doing lumbar punctures on Ebola survivors to see if there was any evidence of viral persistence of Ebola, because we know that this disease can actually cause relapse in the brain. And so I had the unique opportunity to actually look to see if we could find evidence of that. And right next to that, I have a picture of a whole group of people. This is at the NIH Clinical Center. And this was when we were able to bring several different Ebola survivors from Liberia to the NIH for further in-depth evaluation, such as with an MRI scan, EEGs, and things like that that are not available in Monrovia, to see if we could really figure out what sorts of resources were needed in Liberia to help help these survivors. And in the middle, just to kind of point out, or almost in the middle, we have also Dr. Fauci. All the way on the left is Dr. Cliff Lane, who are kind of big players, or at least we're big players in NIAID. So in addition to these projects, and I'm not even going over all of my different projects, but we also have an encephalitis project in Peru that we're trying to get up and running. This picture shows my boss all the way on the left, Dr. Avi Nath, as well as my fellow all the way on the right, Dr. Hanalise Huff, who's a pediatric neurologist, who's been wonderful. And in addition, we also have a multi-site neurodengue project where we're trying to really detail what kind of issues are related to dengue, especially as this is becoming a more widespread and more common disease, affecting many places across the world, including the US and some of our southern states and things like that. So I just wanted to kind of finish up my introduction by saying that I just feel so lucky to be where I am, to be at the NIH, and to get to do what I do, to be able to characterize these lesser studied neurologic aspects of these infectious diseases, in addition to what I do also at the clinical center, which I didn't even go over. So thank you again for having me on this panel.
Thank you, Dr. BU, and thank you for sharing your journey and telling us how you discovered your passion and your interests. And I will now turn it over to Dr. Amareen Mughal. Dr. Mughal is a Statman tenure track investigator at NIH, at the NINDS, and is also the chief of the Neurovascular Research Unit. Dr. Mughal?
Thank you, Dr. Faith Plante, for the introduction, and Open Office for this opportunity. So in next 5 minutes, I thought I'll give you a journey how I reached to the NIH Intramural program, as well as what we do in the Neurovascular Research Unit. So I'm from India. I grew up in a very small town in India, and this is one of the famous river in my small town. And so I got most of my education from India. I did my bachelor's in pharmacy and then master's in pharmacy from India. During my master's, I got opportunity to do a research thesis project where I studied the genotoxic and carcinogenic effect of different anti-cancer drugs. After finishing my master's, I've transitioned to a biopharmaceutical industry and worked there. Although I enjoyed my day-to-day life, I was missing the bench and lab research work. So I decided to pursue my PhD and moved to the United States at the North Dakota State University to pursue my PhD. And yes, indeed, it was pretty cold. On the bright side, I got to spend more time in the lab and finished my PhD. After finishing my PhD, I moved to University of Vermont for my postdoc. And during this postdoc at University of Vermont, I had opportunities to write certain career development award, including NIH K99 and AHA Career Development Award. And by getting those awards, I got an opportunity to transition to a research assistant professor position at University of Vermont. With those career development award, I also had option to during the mentoring phase to look for independent tenure track investigator positions. And when I was doing that, I looked for multiple opportunities, and I also learned about investigator positions at NIH Intramural Research. And in my view, I landed to a very good position for my career stage. I'm a Stadman tenure track investigator, and I started my position in January 2024 this year. So in the neurovascular research unit, which is my research unit, we are interested to understand the communication between neurons and blood vessels, which is known as neurovascular coupling, in the brain homeostasis, including clearance of metabolic byproducts. We are also interested to extend this knowledge to vascular cognitive impairment and dementia, or VCID, using models of small vessel diseases of the brain, as well as cerebral amyloid angiopathy. So to study the neurovascular coupling, we are using a variety of approaches ranging from a single cell to whole animal. And here's an example where these bright green fluorescence you're seeing are the calcium signals which are generated in the vascular endothelial cells of brain cortical vasculature. And these imaging sessions or measurements were performed using a craniotomy-based approach using multiphoton, where we are studying this in mouse models. So this is all very exciting, but I cannot do all of this by myself. So I have opportunity to work with very talented group of individuals in my research unit. And you can learn more about my research program using this QR code or the website link, which is shown up here. And at this point, we are interested to hire postdocs and postdocs. So if you are interested, feel free to reach out. Thank you.
Thank you, Dr. Mughal, for sharing your journey. And we're very happy that you switched from cancer to neurovascular research and that you're here with us today. And our final speaker is Dr. Zaid Khalik, who is a senior investigator in the cellular neurophysiology section in the intramural program at NINDS. I'll turn it over to you, Dr. Khalik.
Thanks so much, Faith. Hi, everyone. It's great to be part of this open stage webinar. So my name is Zaid Khalik. I'm a senior investigator. So I will tell you about my interests and what led me to science, actually. So I think there are a couple types of people. Certainly, some people know from an early stage that they want to be scientists. For me, that definitely wasn't the case. As a youth, I really had like a broad amount of interests. I played basketball in high school. Most of my time actually was dedicated to my instrument. I played a violin, and I continued throughout college. Here's a picture of me when I played at the retirement of our Institute director, Story Landis. And so I had lots of interests, but really my exploration of science is something that I did in my free time. As a kid, I loved physics. I loved chemistry. Here's a popular science book that fed my interests in the topic of physics. So this was something that I sort of really loved when I was a kid, but did it sort of in my free time. when I went to college, actually, I decided that I would try to pursue both my interests in science and music. So I majored in physics. I focused on physical chemistry, statistical physics, and atomic physics. I also took a ton of music classes, played in the college orchestra, and I was in lots of chamber music groups. But it was really after college that I started to focus much more on my science. And so I decided that I wanted to experience, classroom physics was interesting, but I wanted to experience how it was to be an experimental scientist outside the classroom. So I decided to do a postbac, which I definitely am happy that I decided to do. I did it in a physics lab at NYU, where we studied laser cooling and trapping. And really, I didn't, when I decided to do a post-bac fellow, I wasn't thinking like, oh, I want to be a scientist right now. I actually just wanted to just try, working in a lab because I thought it would be the next fun thing to do. I could do it over a couple of years and then, try to figure out what I wanted to do afterward. In the physics lab, I decided that, physics, at least the type that I was doing, was not for me. But I did love the academic science environment. And so for reasons related to my personal life, I ended up in University of Iowa. I followed a spouse out there. And it was there where I did a second postbac in neuroscience. And I really loved it. Coming from physics, many of the principles that I had learned about electricity could all be used in a biological system. And so once I actually learned about action potentials and started doing recordings from hippocampus neurons, I was absolutely sold. I was hooked on this. And so I decided to go to graduate school. I went to graduate school at Northwestern, where I studied the physiology of cerebellar neurons with Indira Raman. I love to work in a laboratory. She taught me how to be a scientist. Incredibly important for my development as a scientist. And here's my picture of my first poster with Indira and Nathan Gowans, who now works at the Allen Brain Institute. I went on to do a postdoctoral study with Bruce Bean, where I studied ion channels and how they interact to generate spontaneous action potential firing in midbrain dopaminergic neurons. Science is an apprenticeship, and with Bruce, I couldn't have picked a better mentor. He taught me, how to approach scientific questions, navigating life within science. All of that is incredibly important, and I am thankful for his mentorship. And so I went on the job market, decided that I did want to be a scientist, professional scientist. I was really lucky enough to get a tenure track position here at the NIH. It's a fantastic place to do research. I love the colleagues. I'm really well supported. It was really a perfect fit for me. So I got tenure in 2020. Now I'm a senior investigator. I'm very happy to be at the NIH. I made the best decision ever. The work in my laboratory actually focuses on dopamine releasing neurons located in the midbrain. We're interested in the function of these cells and behaviors and circuits. Dopaminergic neurons are critical for the reward system, but they also enable movement. And we notice because lack of dopamine leads to Parkinsonism. And so what's important, though, is that in Parkinson's disease, there are a select number of neurons that die. There's selective vulnerability. And the reason for this is actually not known. So the work in our laboratory is actually to find out what's unique about these neurons that tend to die in Parkinson's patients and try to compare them to the neurons that are resilient. And so our goal has been to compare these cells. One observation, which brings me to the research that we specifically do, is that the axons tend to die in these neurons first. That's the first most vulnerable compartment before the cell body. And what's interesting about the neurons that tend to be vulnerable, which is in blue, is that they have these very elaborate axonal architecture. And so one idea is that the branching of these axons may require tremendous amounts of energy and maintenance to survive, and make them more vulnerable to cell death. And so in my laboratory, we've been recording from the axons and trying to compare the activity of these two different cell types. The blue are the vulnerable ones, the red are the resilient axons. They're incredibly small structures. We're able to get pipettes on them, glass electrodes on these, and record the activity. It's really a feat, but I'm thrilled to have be able to recruit talented people to the NIH to do this type of work. Yeah, and so really looking forward to what we'll learn in the next few years.
Hey, thank you, Dr. Khalik. It's interesting how something you explored in your spare time became the focus of your career. Thanks for sharing that with us. So now I will turn it over to Dr. Diamond and all of our panelists for discussion.
Thanks, everyone, for your presentations. They were really interesting. All of you have already, even Hector at a fairly young age, you have some experience in different places, different institutions, different research environments. And I'm just wondering if you might tell us what you think makes the NINDS and the NIH Intramural program unique. What is it that you find here that you might not find other places? You guys can just chime.
That's a very good question, Jeff. Definitely not only so I come from Puerto Rico, the type of not only resources but type of diversity we have in our population is not quite a lot, even though we have some people visiting from the outside. But I would say the opportunity to work besides people all around over the world that meet at the NIH, top of the notch scientific institution, I feel like that's one of the best takes away. In order to collaborate, we all use our strength, and we can rely into each other to be able to promote what is research and provide value to our future. So I would say that's my take. I don't know what the other panelists have to say.
Any other?
I'll jump in.
Yeah.
Sure. I guess from a clinical perspective, since I'm a clinician at heart, is that at the NIH, we really have a unique opportunity to get to study rare diseases. And part of that is, just the way that the NIH is built, the clinical center and things like that. And it's really, just incredible. Also regarding my training, like I was saying, I don't think I could find a place, especially a clinical fellowship, that would have allowed me to do basic research, clinical research, translation, and also, clinical work, but really have the also protected time to do the amount of research I was able to do coming here. So the NIH was a no-brainer. Yeah.
Thank you.
I'd add to that. So when I was looking for opportunities and to start my first independent research program, I was looking for a place which is like where I can grow, as well as diversity is well supported, because it is important for me. And when I came to NIH for my interview, I found that the diversity in expertise, which is present, allowed me to not only focus on my research program, but I can expand in multiple directions. And one thing which I found very unique about NIH, in here, basic scientists and clinicians are working side by side, which is very rare to find. And that gives an opportunity. Like, I'm a basic scientist, but I'm recruited in a clinical department. So I get opportunity to interact with clinicians on a regular basis, which gives me a more broader perspective about the translational aspect of the research which we are doing. And just to plug in for the diversity aspect, although NIH in general is playing an important role with various diversity initiatives, NIH Intramural program is doing several programs which are to support diversity, which I learned. And I'm also part of the Distinguished Scholarship Program, where the goal is to support the diversity of the trainees and improve the diversity in biomedical research, which I found really important. And that got me really interested to join the intramural research.
So I guess I can chime in too. The reason why I actually came to the NIH was a specific reason. I loved doing my experiments when postdoc, and that's just a feature of science that I really didn't want to give up. And here, it was very clear that I'd be able to continue doing my research and do my own experiments actually. And so, and what enables this is that the structure is that we are given a grant, and we start off with a grant. We have to justify the grant. So the grant funding structure is something that actually frees up your time and actually the result of that is actually they're not huge labs, they're pretty small labs because of the grant structure here. But it was fine for me because the type of research that we do is very sort of small group oriented. I do electrophysiology, we have rigs. The groups are not that big anyway, and I could actually do my own research. So it frees up time for a lot of things. It frees up time for mentoring, for interacting with the people in the laboratory, everything that I liked about research with small groups, clear interaction with folks is something that I thought that I could get at the NIH.
Thank you. So Zaid, I wanted to ask each of you, and maybe we can start with you since you were just talking about this, about the resources that are available. I think people would be interested to know, like from Hector, whether he gets the resources he needs to do work at a very early stage, whether Jean gets the things that she needs to do good clinical work. You touched on the grant structure. How is the support for your lab determined within the intramural program? I think that's something a lot of people don't understand.
I see. Yeah. So we are when we are recruited to the NIH, we get a startup can, a pile of not can. People don't know what can means. We get a startup
A pile of money, you were going to
budget, startup budget. And we also get a yearly budget. And so the startup is for, buy big equipment, things that get your lab started. Everyone will know this. But every few years, we actually have to go in front of a board of scientific counselors. We write a grant style document, which has future directions in it, as well as summary of what we've done over the last few years. We give the written document to the board of scientific counselors, which comprise about how many, 15 or 18 people. And so they read it and they evaluate our progress, and we are and that's the point at which we either ask for money or we defend what we've done with the money we've been given, and so that's our sort of grant-like process, and I think it's a fantastic process, and this is how our grant sort of cycle is. It's about three years for tenure trackers and every 4 years for senior investigators, and I'm very happy with the process as it is.
So Amreen, thank you, Zaid. Amreen, you haven't had the pleasure of going through a board of scientific counselor review yet, and I say pleasure in quotes. I think Zaid would agree that it's a bit of a nerve-wracking experience. There's a lot of pressure there, but how do you feel do you feel like you've gotten the resources you've needed to start off your lab on the right foot?
Thank you, Jeff. That's a great question. Yes, I've been learning about the committee review, but I'm not yet there. So when I started my position, I've given plenty of resources, including I've given an opportunity to establish a mentoring committee who has been really helpful. The committee is not helping me only with the administrative question which I have for establishing a lab or how to get through day-to-day process, but they are also directing me how to recruit trainees, what are the different opportunities available for early career investigator. So that's really great resource. I also learned about senior advisor for faculty development, which is also is developed to answer any questions which early career investigator or tenure track investigators have related to the tenure process or any of the rules and regulation associated with that. There are plenty of resources available from Office of Training and Education, which I've been using related to recruiting postbacks and postdocs. Also, there is a Women Faculty Advisory Committee, which has like a peer mentoring program. So I'm still evaluating different resources which is available for me, but I think I'm well supported in terms of the resources which a early career investigator need to establish a good research program.
Thanks, Amreen. John, you have you have a kind of a unique existence as a staff clinician here with this very outward facing program. Has that been a challenge, or have you gotten the resources you need to do that?
Yeah, I'm in a unique position. I think I have a little bit of funding, which normally staff clinicians do not, because I am a clinical entity here at the NIH. And so I do see a lot of patients here at the clinical center. But in addition, I have also my research, which every clinician who is here participates in research, because every patient who is seen here at the NIH is part of research. But yeah, I think, kind of through some issue with the pandemic, I kind of ended up with a little bit of my own funding. And it's been really great to have that, to be able to kind of act as a more independent researcher, to be able to travel to Liberia often to set up research projects and things like that with my own funding, which of course is actually still taken care of by my, it's still under my boss's purview, but I like I said, I just feel incredibly, lucky to get to do what I do. And I think it is important because it's, even though we do, global work, it is things that these things are things that come back to the, patients who are in the United States and things like that, and to the global community. So anyway.
Thanks. Hector, how was it for you? You come in at a very early stage of your career. Did they let you touch stuff in the lab? People don't let me touch stuff anymore in the lab. They're afraid I'm going to break something, but
it's actually pretty funny, especially during my interview. One of the things I was able to provide, thanks to my experience by shadowing with the ophthalmologist, I made a question to my PI during the interview that, are we foreseeing the synergy between or correlation between the eyes and the brain images? And actually back then, the OCT scan they had back then was damaged. And they were like, if you join our lab, Hector, we'll make sure that we can get an OCT that is running. And I was amazed about not only the opportunity I was going to be able to start my project towards what I explained earlier, retina and cortical lesions in multiple sclerosis patients. So I was amazed because in Puerto Rico, the budget, or sometimes whenever you're working with, let's say, a vial with a solution, it will be hard, or if you spill something, you will get, let's say, there could be some consequences to that because we have very, very limited budget and not so many resources within the lab. And also the NIH, like I said earlier, provided resources not only in the lab, but outside the lab with not only the OITE program that helped through the preparation for the next step. In my situation is medical school, but there's a lot of help for graduate students, for people who are preparing towards the next journey, and also a lot of classes and continued education that you can take within the NIH thanks to the resources. So I would say it's amazing that there's in lab and outside the lab resources that you can be benefited as well for the wellness advisor. So yeah.
So Hector, when you're in college and you're thinking about doing a postbac, right, and you go onto the website, there's 1,200 labs.
Yeah.
I mean, that's really kind of overwhelming, isn't it? I mean, how is your experience in navigating that and reaching out? It's also intimidating to reach out to scientists to ask them about the work in their lab. How is that for you?
Well, it definitely is a little bit scaring at the beginning, but since my experiences doing research, I was already with a specific topic I wanted to investigate and do research. So I only refer myself to the NINDS, and in the only at the NINDS there were many labs, but then I was with the filters and narrowed down on what topics I would like to expand into my future or where I could see myself working besides with other scientists and other fellows. So I reached over I would say over 40 different PIs. And I had this list, it's about be determined of maybe what area you want to go. And that's how I ended up in neuroscience. And it's been a really good opportunity. But I would say it took quite a bit of help. But also I received help from Angel de la Cruz, who's my current mentor. He reached out and he also helped me through the process of, all right, so there's this many labs, and I would say I'm not afraid to ask and seek for help when it's needed. So I believe we need to make questions when we're lost or reach out to someone that might have more experience, or you'll end up be getting referred to someone else. So I would say that's very helpful.
That's good. That's a good approach. So you narrowed it down to some labs that you figured you would be interested in. Can you give some pro tips about it? Did you find it more effective to express specific interests in the work that someone was doing when you contacted them?
Of course, yeah. It's very so what I did is I finished my application through the website. I got my letters of recommendation. I put all the information. It was the requirements, obviously, for the application. And after that, I began the journey of emailing and doing a very unique email to every PI I was interested in working on. So that's what I did. I read about the background. You read about their work, see the latest publications, and you like build this background of maybe what things you might be working on, and then you'll be like, okay, is that something is really interesting? Is that something I really see myself? And that's what I did, and got lucky enough, got a couple of interviews, and then I had to narrate it down to the lab that I see myself more fitable.
Yeah. And Zaid and Amreen is when we apply for faculty jobs, that's not you look at other places. Was it super different applying for a faculty job here versus a university, or was it similar? How did it compare?
I can go first. So I applied through Stadman Tenure Track Investigator Search, which is an NIH-wide search, happens every fall, early fall. And the application process was very similar to what I've been doing for other universities. There were like some documents which were including your research statement, your resume or CV, as well as like a diversity statement. Like after that, the process was a little different than other places. I was selected as a semi-finalist, and then the process depends if any institute is hiring or interested in the research program which you propose. The goal of this Statman investigator search is to recruit early career investigators who are interested to have a research program in high risk, high reward manner. So I was the shortlisted candidate for that, but I also applied for the stroke branch search, which was happening separately, which was very similar to any of the research universities job search, where you come for interview, you have 2 rounds of interviews and give a research talk, a chalk talk. So there were some similarities, but there were some differences. And I would be happy to expand on that. If anyone is interested, they can reach out to me. Thank you.
Thanks.
I guess really quick, I'd say just you're being considered by multiple institutes when you apply here, and so the application is the same, but you are being considered by multiple institutes. And I just want to go back to something that Hector said that's so important, which is that people are applying to different laboratories. If you read the research in the laboratory and you write a letter that puts yourself and your interests in that laboratory's research, you will be so much more your application will stand out to that laboratory. For all of the postdocs and even graduate students who are interested in the laboratories, definitely, write in the letter to your PI, you know, what you like about that laboratory.
Yeah, that's a good tip. John, I'm curious, you know, our clinical center here is amazing. There's a lot of really significant differences. I mean, there's still a lot of very committed physicians tending to the care of patients, but there are a lot of differences. How do you think the practice of neurology is different in our clinical center compared to an academic medical center like Hopkins, for example?
Yeah, it's a really unique place. So the main difference, I think, from other other academic hospitals is the fact that the clinical center is solely a research hospital. So every patient who is at the clinical center is under some sort of a research protocol. Sometimes they're getting unique care, experimental therapies, even if they have common diseases. Sometimes they are under a research protocol for a rare disease. You know, they might be like one of 20 people in the world known to have this issue. And so, you know, people are studying it to try to figure out if there's any way to treat it. And then also natural history studies of both common and rare diseases. So whenever I practice, typically all the patients I see are very, very complicated. They've probably seen 20, sometimes they've seen 20 doctors who didn't know what they had, and they come to the NIH because we actually have seen this disease before. And then another thing is that we don't have an emergency room, so you can't just come here, you know, and be seen. You do have to be filtered in some way. And sometimes you're referred to by your primary care physician because you have a rare disease or, you know, a unique presentation of it, or you qualify for an experimental therapy, something like that. Or there are also, you know, ways to find out if you or your loved one or somebody has a disease, you know, whether or not there are any kind of trials going on at the NIH. And that's very easy to find out on, you know, the NIH websites and also clinicaltrials.gov. So yeah, it's a fascinating place, and we have lots of resources, and also the patients do not get billed for their care here. So that's another very different aspect. So I don't have to do any billing, which is fabulous.
Okay, thank you.
Thanks.
Well, thanks everyone for your input. I better turn it back over to Faith and Anna. I'm not sure, Anna. We've been going for a while. There's a lot of really interesting information that I think that you guys contributed.
Absolutely. Thank you so much to all our panelists for joining us today. This is going to be recorded. Please do share it. I will send an email afterwards with all the resources, with the slides and everything. And this is really meant to kind of demystify the intramural program for you all. So hopefully now you have a face to a name, and you can reach out and ask more questions afterwards. So thank you all so much. I'll be in touch. Take care. Bye.
Thank you.